So we stopped here last week when we were doing our quiz. So we're just going to continue here in the first hour, I believe, or less, to complete the quiz that we had. And I had the liberty to also add some contents that we started or we finished on session 1 last week. That will be like the questions about your deductive and inductive reasoning. And please also do research and do inquire about the previous past question papers. That will also be helpful to you on your studies, so that you will be able to test yourself and be able to help yourself with regards to how will you be able to answer some questions in the exam. I think we did this one about where they were saying during a biology lab, you are asked to look at a prokaryotic and eukaryotic cell under a microscope. Remember, I just tell you, if one person could give you a 1 rand every time I say prokaryotic and eukaryotic cell, by the time we are done with these tutorial sessions, you will be a millionaire because we're going to talk a lot about prokaryotic and eukaryotic cells under the microscope, and I document what components they both share in common. Which of the following is not a feature that all the cells have in common, those prokaryotic and eukaryotic cells? And I think the answer was B, where they say though some of the eukaryotic cells have cell wall, such as plant cell, not all do. All animal cells do have cell wall, and therefore not all cells share these characteristics. So this is what we're going to be continuing today. Yes. So here they say, Stacy, yes, are we ready? Just show me with your thumb. Are we ready to tackle today? Okay, cool. Stacy is a field biologist. So the field is in any career. So if they talk about journalists, when they tell you we are going to the field, it means maybe there is a story. Maybe there is a president who is coming and he's doing door to door. You know, now it's election season. So the president will be going to house, door to door. So there has to be the journalist who will say, we're going to the field. When they're talking about the field, they're talking about we are going directly where the story is happening. So there will be your anchors, who most of the time we see on SABC or on whatever, on each where they see you see them on the TV, but they will also have some field journalists who are following the story right at the core of what is happening. Maybe it's a school, maybe there's an incident, maybe something happened, but those people who are there, they're called a field. So even in biology, so a biologist could be somebody who's staying in the lab, who's doing something, or a bioinformatician who's sitting analyzing the results. But the one who's the field is the one who's collecting the data, who's going and to say, most of the time you'll hear biologists said we're going to the field, or you'll come across it when you're on your second year or third year or in your honors, definitely, where the supervisor will say, you're going to be doing this as a project, you're going to be doing this, but you're going to have to go to the field. That's when you're going to be going to collect the data. So they say Stacy is a field biologist collecting water samples and has recently discovered a new unicellular organism in a sample she collected. Stacy has classified this organism as being eukaryotic, okay? And let me, with my mouse here. And which of the following could not be a feature of the organism she discovered? Let's go. You know the rules, just unmute yourself and give us an answer. Yes.
Is the answer B?
Okay. Anybody else?
Oh, sorry, no, it's the eukaryotic, so it has organelles. So the answer is okay. No, I'm
Yeah,
not sure. I thought it was
you're not
eukaryotic. So
sure. Yeah, but I like your approach. Your approach to say, what eukaryotic has, what features does it eukaryotic have, what characteristic does the eukaryotic have, and then we are looking at the feature that is not eukaryotic.
So what's a capsule, if I may ask? I haven't seen anything about a capsule.
It's I'll answer that. I'm noting the question. Yes, I will answer you. Anybody else? We just need a minimum of three opinions or two.
Morning, sir.
Morning, how are you?
I'm good, and you?
I'm good.
I think answer is D.
And the answer is D. Do you mind if I may ask why? I guess you do. Anybody else?
It's A.
Somebody says it's A. The gents, where are the gents? I still said that Real Madrid beat Manchester City. I know this generation love Manchester City. Where are the gents? Any
Can I say the
response?
answer?
Yeah, sure.
There.
Sure.
Morning, how are you?
I'm good, yourself?
I also I also think the answer is D.
You also think the answer is D. May I also ask why do you think it's D?
I think it is because the question said
Mm-hmm.
Stacy is a biologist collecting water samples and has recently discovered a new unicellular organism in a sample she collected.
Mm-hmm.
Stacy has classified this organism as being eukaryotic, which of the following could not be a feature of the organism she discovered? If we talk about diameter, don't we talk about the half of a circle in mathematics?
Yeah, but the cell diameter is just to be honest, just a size. They just you just have to be like between the sizes. Which one is? If you go to those, if you go to those cell sizes, which one is it for? Does a eukaryotic fall within those ranges or not? If you can just go to one of those, can we go for the answer?
I think it's C.
You think it's what?
I think it's C, a capsule.
Okay.
Because eukaryotic cells, they do have a nucleus. They are membrane bound. They have membrane bound organelles inside their cells, and their diameter, if I'm not mistaken, it ranges from 20 to 100. Can range from 20 to 100. What's that one? Is it micrometers?
Mm.
So I
And
think C one.
and that will answer the question for the student who said, what is a capsule? Because you are correct. The answer will be C, because eukaryotic. Go back, we'll also reveal that. Go back to the comparison between eukaryotic cells and prokaryotic cells. Eukaryotic cells do not have capsule. What is capsule? There was the first person who asked. She asked, what is a capsule? Capsule are made of polysaccharides and are often encase bacteria, which are prokaryotic cells. Though there will be features which are like a tiny little details, and for you guys to pay attention to that, you're gonna have to thoroughly be aware about the differences between eukaryotic and prokaryotic cells and those characters. Remember when I said, even when you're looking at a diagram, please study the diagram or label it. What are the functions of those labelings? And then you'll be able to go. And I'm sure, and I know, especially multiple choices questions could look very similar and look like all the answers are correct, or the answers that you're going for, it seems like it's really, really the one, but when it's really not. All right, we move. And which part do all prokaryotic and eukaryotic cells have? I think we did this last week. I might be repeating a question here, but we'll see. If it was here last week, then obviously you should be able to shoot the answer fast. Which part do all, they're asking about something which is both present here.
The answer is D.
All right. Okay. Anybody else?
The answer
I
is
concur.
I didn't get that. What was that?
I said I concur with D also.
You concur.
but-
Okay. Okay. We do have two unanimous now.
I will say it's A.
Okay. There's a different one. Anybody who wanna?
I think it's C.
We have different one now. C. There's a C as well. We got two for who said is D, and one who said is A, and one who said is B. Do we have the final arbiter? Last judge?
I think the answer is A.
Oh, we got two who said it's A. Okay, let's see. The answer is D. All prokaryotic cells have plasma membrane. You know what? Let me do this. I know you can see my screen now. I just wanna, there's a slide which I was gonna bring it a bit later, but I just wanna do it now. That way we will be able to don't forget to put your student numbers on the chat box. Yes. And we are back. What is my laptop doing? Yes. So the answer is D, right? So just want you to see here the differences between prokaryotic and eukaryotic cells. So this will be one of the person who was writing an assignment. Remember when we were talking about how you can write and just scan it and how you can just do. So prokaryotics are very minute in size. I can read this handwriting, yes, but it's fairly large in size. And the nuclear region, nucleate not surrounded by a nuclear membrane, nuclear material surrounded by nuclear membrane, single chromosome present, and what is my laptop doing now? More than one chromosome present in the eukaryotic cells, nucleus are absent. On the prokaryotic, nucleus are present in the eukaryotic, and membrane bound cell organelles are present in the prokaryotic, and membrane bound organelles are also present in eukaryotic. So you can see which organelles are present, or which functions are present in both eukaryotic and which ones are absent in them. And the cell division by fusion or budding or mitosis, no mitosis. Then in the eukaryotic, cell division by mitosis or meiosis. in eukaryotic cells. The division or production in eukaryotic cells occurs by means of mitosis or meiosis, but in prokaryotic cells we have fission or budding. Yo, what's happening? My laptop is not responding. Come on, what's happening? Can you guys still see my screen? Sorry about that, guys. Okay, cool. Thank you. Then, okay. Okay. Just hold on a second. I don't know what I did. My laptop is not on my side. My screen is not moving. Okay, there. Is the screen moving now from you guys? Can you see? Okay, then when it comes to the other prokaryotic differences, you see here also, we see the number of chromosomes, cell type. This is the differentiation that I will prefer that you guys have, so that you can be, so we see this question here. Come on, what is happening today? Don't do this to me. Sorry, guys. I think this laptop doesn't want me to connect with the mouth. I'll just have to navigate using my touchpad. Sorry about that. Believe you or not, I'm trying to move my slides, but nothing is happening. So while we are waiting for you to respond, so you'll see here there's a number of chromosomes, cell type, mitochondria, chloroplast, cell size, structural complexity, DNA found in the region, membrane enclosed organelles, lysosome and perisosome. We did talk about this in previous sessions. Endoplasmic reticulum, Golgi apparatus, permeability of nuclear membrane, plasma membrane. There was a plasma membrane now in the previous question. This is the question that I wanted us to go back to. Then cell division, and then ribosomes. So this usually helps with the answering of the questions. So this is the question that we were doing. What part do all prokaryotic cells have? Or there probably are more, but your examiner will probably give one, so that if you get, unless, and you must be prepared that there will be questions which are not as clear as this now, which maybe he may put all the question could say, which part do all prokaryotic and eukaryotic cells have? And he put all of them, which are the same. And he will have a fifth option, which says all of the above. So you must be ready for questions like that. So here in this case, we were talking about plasma membrane. And if you check here, you can see there's plasma membrane here, which is present and present. And sometimes will be a question which says which feature is present in the eukaryotic cell, but is not present in the prokaryotic cell. So please do have this structure, like not only in a question of prokaryotic cells only, but even in different questions. Okay, what do prokaryotic? We agreed it's plasma membrane. Another one is, how does a typical prokaryotic cell compare in size to a eukaryotic cell? I'm sure you just saw a slide like that. It's just that I removed that one, those slides of comparison, but you could have just on what it says. It is smaller in size by a factor of 1 million. It is larger in, this is prokaryotic. It's prokaryotic cell to eukaryotic cell, not eukaryotic to prokaryotic cell. So you're gonna have to. It's not just a differences, but it's a different from a prokaryotic to eukaryotic cell. Or C, is it smaller in size by a factor of 100, or is just similar in size? And you need to also have a strategy to eliminate wrong answers in multiple questions. But anyone want to give it a try? Hope my slides are visible now. Yeah.
For this one, I think we can just eliminate the larger factor by, it is larger in size by factor 10, and it is similar in size. So the answer is between A and C, but I'm not sure. I'll just say C.
All right. I like that scientific guess. Yeah. Anybody else?
I'll also go with C.
She will also go with C. I'm a bit worried about ladies. Do not bear with the gents, right? There was a soccer in the middle of the week. There's a team that is loved very much in South Africa called Manchester City. It was beaten. So it was the last option where people will say, "I will." Since Chiefs is losing, we will hide under Manchester City. So I'm assuming all the gents are depressed because it's only the ladies, guys. I'm worried. Where are the gents? Anybody else who want to give? We do have C. Two people are studied C. Anybody going in?
I'll go with A.
You go with A. It's smaller by it is similar in size by a factor of 1 million.
Yes.
1 million. Anybody else?
Sorry, I would go with C.
You go with C.
Yeah.
Good morning. How are you?
Good. I'll also consider going with C.
You will also consider going with C. Thank you so much. Yeah, you are right, guys.
I'm going with C.
Yes, and the answer is C. With prokaryotic cells are significantly smaller than eukaryotic cells. And let's go to the next one, guys. It's drag the words. We did a question like this in the previous session last week, where you have to drag the words into the correct boxes to complete the following statement. It says prokaryotic cells are surrounded by a number 1 membrane and have DNA, cytoplasm, and like eukaryotic cells. They also have number 3 walls and may have cell, which is number 4. And there's a prokaryotic have a number 5 large chromosome that is surrounded by number 6 membrane. Prokaryotic may have number 7 for motility, which is 8 for conjugation, and for adhesion to surfaces. So you're going to have to drag and place these into those cells. So you're just going to have to say 1 and you tell me which is either capsule, B or single, or C spily, or D nuclear, or E flagella, or F cell, or G ribosomes, or H fimbriae, or I, which is plasma. Let's go.
For number 1, prokaryotic cells are surrounded by a plasma membrane.
Okay. Anybody else for number 1?
I'm sorry. She said a what?
Oh, she said a membrane. I think.
Plasma.
Okay, what do you
Plasma.
think?
Okay. Plasma membrane.
It's plasma membrane. Anyone else? We're not gonna dwell much into one here because there are a lot. Anyone?
I think it's F.
It's F, which is cell. Okay. Let's just unmute yourself if you wanna say something. Yeah.
Say,
Okay.
I think it's nuclear.
It's nuclear. All right. Okay. All right. I think I'm gonna.
The correct one is I.
It's
I think it's I.
I. Okay.
Yes, number one is I, plasma.
Number one is I. Okay. Let's see the verdict.
Oh.
And yes. So the prokaryotic are surrounded by a plasma membrane. Also, look at the, okay, I will say this when we're done. Plasma membrane and have cytoplasm. And what is number two?
Ribosomes.
Anybody else? Which is the last one? Who's the last one for this one?
Ribosome.
Ribosome. Okay, let's see. And yes,
Ribosomes.
it's ribosome for cytoplasm and ribosomes, like eukaryotic cell. They also have, what is number three? Walls. This is simple. Come. This will be cell. This will be cell walls, and may have a cell. This is also simple.
Membrane.
Anybody
A.
else?
Membrane.
Anyone else?
They also have a
A.
Somebody says
Letter
A.
A.
Yes.
C.
Anybody else? There's somebody who said C.
Capsule.
Okay, we do have two who said A. All right, let me just give the answer is yes, and the answer is indeed A. Capsule. Then I think somebody needs to mute themselves. Yeah. Then prokaryotic have what is 5?
Single, maybe. Not sure.
Yeah,
Okay.
single.
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 Let's see.
Answer
Yes.
single.
Then prokaryotic have a single large chromosome that is not surrounded by, what is this one?
D.
I think
Nuclear.
this one is simple. Yes, the nuclear membrane. Prokaryotic may have, what is the function? These are the functions now. They're giving you the function to say what is the function of number 7. It's for what? For motility, which is gonna be what?
Flagella.
I think is letter H. The answer is letter
Flagella.
H.
Was flagella.
There is a guy with a flagella. Let's see. Exactly. It's flagella for mobility, for motility. What is for conjugation? What is number 8, which is for conjugation?
Fibria. Fimbria.
Anyone else?
C.
Pili.
I would use flagella because I know that pili is for adhesion. So by process of elimination, I'm thinking the other one works for conjugation.
Which one is that? I'm sorry, I didn't get that.
H.
Fimbria. I don't know. H. Yeah, I don't know how to pronounce it properly.
Might be wrong, but C.
Single.
Yeah.
Again. Also, it repeats.
So I made a mistake, isn't it?
Because B5 is also single.
Which one is it's left? We will figure it out.
And C.
Oh, no.
H.
Which one is left?
H and C.
H and C.
Pila,
H and C.
And the correct answer, I think, for number 8 is letter H.
Yeah, I think H also. Because single, if we say for motility single for conjugation, it also does not make a lot of sense.
Perfect. And what are we saying the 9 is? For
I'm saying
addition.
9 is pili.
I say it's C.
Oh, it's the other way around.
But you are right about 8 though. The 8, there's a mistake. I literally put a mistake for deliberately for number 8. Which number 8 is what? It should be what? Because the omission. So there are some questions where you won't have like, if for some other way around for some reason, there's another one that you are not sure of. But you're gonna have to use the law of omission to say the elimination to say the correct one is this one and the correct one is that. So by the law of omission, see what will be number 8. Number 8 should not be single. It should be what?
Pili.
Pila. Yeah. So when you get these slides, please do change this. You will?
Okay.
Let me change them now so that somebody does not say, no, Morunga, you're the one who you gave us a wrong slide. Cool. Fine, right? Okay, let's move on to the next one. So here you see this diagram. It says, please ignore the numbering. The last time I did this, my laptop froze. Hope it won't. Yeah, so please ignore the number in figure. Just take it as figure. So this is the scientific method consisting of series of well-defined steps. If a hypothesis is not supported by experimental data, one can propose a new hypothesis. So here we're just saying, please pay attention to this diagram and have this diagram with yourself in a very good place where you can be able to when you're studying and have to, this is gonna be like your frame, your structure, like your template in which you can be able to answer every question. So you make an observation, ask a question, and form a hypothesis. When they say if a hypothesis is not supported by an experimental data, one can propose a new hypothesis. That's why they say try again. So make a prediction based on the hypothesis, do an experiment to test the prediction, analyze the results. So when you analyze your results should be able to support your hypothesis or not support your hypothesis. Then you report the results. But if your results, the analysis of your results does not support your hypothesis, then you try again. What is a hypothesis again? Just a hypothetical, just a passing question. What is a hypothesis? Anyone?
What you believe the result of the investigation will be.
All right. Cool. Yeah. And let's go. So from this diagram here, I wish I did put the diagram somewhere closer, but have this diagram in mind. Then from the figure behind, in the example, scientific method is used to solve an everyday problem. Match the scientific method steps. Number the items, so it will be number 1, 2, 3, 4, 5, 6. This will be your observation, question, hypothesis, experiment, results, and the analysis and reporting of the results. And observation, question, hypothesis, prediction, experiment results. So with the process of solving the problem with the letter items here on your right hand, based on the results of the experiment, what is the hypothesis correct? If it's incorrect, propose something of an alternative. We'll also delve into this question, but for the time being, we need to match. We need to match. And also, you're gonna have to answer this question, because based on the results of this experiment, because it's an experiment. This is an experiment. Here, this is just a scientific methods which they are using to arrive at those results. So we'll also need to answer the question which says, is this hypothesis correct? There's something wrong with the electrical outlet. If something is wrong with this outlet, my coffee maker also doesn't work. I think I made a mistake. I should have just put the whole thing on top here to explain what is happening, but nevertheless, you can still see my toaster doesn't toast bread. I plug my coffee maker into the outlet. My coffee maker works. Why doesn't my toaster work? So you're gonna have to match and say whether if it's an observation, if it's a question, if it's a hypothesis, if it's a prediction, if it's an experiment, if it's a result. Let's go. And unfortunately, here we're gonna start with a 1 because I arrange my answers in that order. But when you're answering your questions, you can start with anything that you.
Sir.
Yes.
Morning, sir.
Good morning. How are you?
I'm good, and yourself?
I'm good.
I'm good. Sir, I think the observation here is my toaster doesn't toast my bread.
Okay.
That's what I'm observing.
That's what you have observed. That's what you have observed, because you want to toast your bread. When you want to toast your bread, you're making an observation. Now it's my toaster is not toasting the bread. That's an observation. Thank you. That's correct. Number 2, anybody who want to go for the question?
The question,
Okay,
I think,
question.
is why doesn't my toaster work? Sorry.
No, it's okay. Anybody else?
Good morning. The question is F. Why doesn't my toaster work?
I think this is pretty much obvious. Why doesn't my toaster work? That would be F, yes. And the hypothesis, which is the entirety of the question.
The hypothesis, I think, will have to be B.
Okay. If something is wrong with the outlet, my coffee also won't work. Anybody else? Do you agree with her?
There is something wrong with electric outlet.
So yours will be A. Anybody else?
I'll also go with A.
Yeah, it's A. It's A.
So you made an observation to say, okay, my toaster doesn't toast my bread. I want to toast my bread. It's morning. It's 6 a.m. I have this tutorial session with Murunga at 8. And then question 2, what is the question? Why is F? Why doesn't my toaster work? That's the question. And then most of you are saying the hypothesis should be A, right? And you say there is something wrong with electrical outlet. So what is a hypothesis? We mentioned it and we said in the previous session, or the simplicity is that it's your outcome, your predicted outcome, the prediction of the outcome which you're thinking this could be the results of the experiment, or this is the results of the independent which is being caused by the issue or the problem with the issue. So you're saying there is something wrong with electrical outlet. Let's see the verdict. And okay, you are correct. Then what is the prediction?
Prediction is B. If something is wrong with the outlet, my coffee maker won't work.
Anybody else who concur with that, or anybody with a different answer?
The answer is B.
Yeah.
I
also think the answer is B.
Okay, let's see. And you are right. Didn't I make it tricky here? Okay, it's B, prediction. If something is wrong with the outlet, my coffee maker also won't work when I plug into it. So the experiment is it's what you do. So the experiment so the experiment is what you do. So the experiment is what you do to test your hypothesis, to test your prediction in some way. And most of you are saying it's D. So the experiment will be B. All right. Okay. Congratulations, guys. Which means the answer for the results is
That works.
my coffee.
My coffee maker.
My coffee. Okay. There's based on the results, is the hypothesis correct? If it's incorrect, propose some alternatives. Okay, what was the hypothesis? Let's check.
My toaster does my bread.
No, the
There is
hypothesis.
wrong.
This is so
There
Electrical
is
outlet.
something wrong with electrical outlet. And what did they do on the experiment? It's D, Mike.
I plugged my coffee maker into the outlets.
The outlet. So
I think the hypothesis is not correct because it's talking about that there is something wrong with the electrical outlet, but then with our results, we see that the coffee maker actually does work.
And
The outlet is the problem is not the electrical outlet. The problem is the toaster.
So we need a hypothesis.
Yes.
The hypothesis is correct, though.
No, like based on the results of the experiment, it's not correct, the hypothesis. Because the results say if the coffee maker works, it means the electrical outlet also works, right? And there we were saying with our hypothesis, there's something wrong with the electrical outlet.
Which means
the
okay.
coffee maker
Okay, let's just have an understanding right here. So an electrical outlet will be what? Will be your
plug.
Your what you plugged on, yes. What you plugged on, yeah. So he is saying in his hypothesis there is something wrong with the electrical outlet. So to prove that, what did he do? He did an experiment. What is experiment is D. He plugged the coffee into the outlet. And what was the result? The coffee maker was working, which means the reason for his observation, because his observation was my toaster doesn't toast bread. Okay, we made a mistake here. It's supposed to be my coffee maker, isn't it? So it means maybe it was supposed to have been no, my toaster, there's something wrong with his toaster, not the electrical outlet.
Exactly.
So which means his toaster it's what it's yeah, need to buy a new toaster, guys. Is that clear? Yes.
Yes, it's clear. May I please ask something?
Yes.
How do we properly separate between a hypothesis and a prediction? I think I struggle a
That's
bit there.
That's a good question. That's a good question. In that's a good question. I want to use this in this context, but I'm gonna struggle. But the hypothesis you can you can test it. You can test it, but after you can just predict your results that you're gonna formulate on the basis of your hypothesis. But the long and it is that the hypothesis has to be tested. You can test the hypothesis, and if your hypothesis cannot be proven by your results, you can just redo another hypothesis, or you can you can propose a new hypothesis, not redo a hypothesis, but a prediction. You predict the results which you can do, and the prediction is not to be a blind prediction. It has to be an informed prediction based on the previous results or based on the related literature from previous work which has been done related to the topic that you are discussing. But the prediction and hypothesis can sound very similar, but a hypothesis you can test it. Prediction you cannot test it. You can just predict based on the information or related to what you. So let's say it's elections, right? And no, let me not make an example with election because I might offend people. It's a let's say it's results, it's metric results, and the government want to disperse resources to through provinces. Gauteng province, Limpopo province, Eastern Cape, and so they use results from 2023 metric results. They check or Limpopo got number 1, Western Cape got number last. I'm just making an example. I'm not saying this thing.
Sorry, we can't hear you. Your mic is muted.
You and now? Sorry about that. Unmute me. Can you hear me now?
Yes, we can hear you.
Yes, we can hear you now.
But you guys can still see my slides, right? I see even in the chat here. Sorry, guys, I wasn't checking. Yeah. So as I was still saying, the prediction, it's based. You predict your hypothesis. You make a hypothesis which can be tested, and which can be tested by an experiment, or which can be supported by an experiment. And that hypothesis, you predict a hypothesis. You don't hypothesize a prediction. So you don't make an observation, right? When you make an observation, you don't make an observation and say, in this, let's predict. No, you don't. Your toaster is not working, and you don't say, let me predict why my toaster doesn't work. You hypothesize. So, you make a hypothesis, and then you predict your hypothesis. Is that clear?
Yes, sir, it is. Thank you so much.
All right.
It sounds confusing, man.
It's confusing. Excuse me?
Sorry, which chapter is this? Because of I don't know. my first day today. So I just
It's
want to
your
know which
first.
chapter.
So this will be your. Anybody who wanna assist him? It's his first day. Which chapter are we in? We're in the themes of biology. Theme 1. We were talking about the phylogeny, the themes of biology. It's theme 1. Do you have your tutorial letter?
Yeah, I do.
You do, yeah. We're in the first chapter. We did chapter 1 on the first session, which was 2 weeks ago, and we concluded it last week. And then we started with the quiz. We're just doing quizzing previous question papers and potential questions which could come into your exam or in your assignments. So just here we're just doing this quiz. We're quizzing each other. We're bringing the questions, and then if you think you know, you can raise your hand. If you don't know, you can take notes. And yeah.
All right.
Yeah. I'm sorry, man. But you started what? You started with the at 8, or you just log in now?
I just like you know. I
You
just
just
came here
log in
for
now.
now.
Okay, no, don't worry. Just give it a 10 minutes or so. It will make sense. Don't worry.
Okay.
Yeah. Sorry about that, man. And based on the results. So this is basically what we were doing, right? And yeah, I didn't. So ignore these slides. I didn't swap them. I didn't correct them here. Yeah. And then next question is, which of the following is not an example of a phenomenon that can be tested using scientific inquiry? So my brew here, who was just saying he's confused. We did also a topic about scientific inquiry, also, where they talk about research, where you talk about themes of biology, definition, what is zoology, what is biochemistry, and all of that. In your study materials, you'll be able to see. So here, this question, which is which of the following is not a phenomena, or it's not something that can be tested. We said if something have to be tested, your A will be, what do your fingers prune when you're in the bathtub for too long? So we remember when we talk in science, you have to be able to test. It has to be tested. It has to be tested. You need to test it, and you need to make an hypothesis. You can make an hypothesis. You can also make and arrive at an informed decision and also make an experiment. B, what causes grass to die after a dog urinates on it? How many ghosts are haunting this museum? Okay, somebody needs to mute themselves. Yes, thank you. Why do plants grow towards a source of light?
C.
I think it's C.
Okay. Okay, I think most of us
It's
agree that it's C. Why? What do you think it's C?
I think that it's because ghosts cannot be tested, and they cannot be tested. Nobody can prove that they exist.
Okay, anyone else? It is C.
So it's because in the scientific phenomena there's no such thing as ghosts. They cannot be tested, they cannot be seen, and anything that cannot be seen with the senses is therefore non-existent.
Yeah. So we don't want to step into anybody's religion or anybody what they believe in, but it's just that science is just, in science you need to prove. So in science, what you do in terms of experiment, somebody else has to come and do the very same thing that you have done and arrive at a certain position. It may not be the same results that you can arrive at, but you need to have a path or a method which I can prove or I can arrive at a results. So yes, supernatural phenomena such as ghosts and spirits are not that we don't believe it or not that you don't believe in it. Yes, you can believe it, but scientifically they cannot be tested. That's the issue. Science is about testing. We need to test. We need to test if this is right, and then we go. So that's why you cannot go to court and said, "This is my child," and I say, "Okay, what do you mean this is my child? This is my child. This is my child spiritually. This is my spiritual child. I'm his spiritual father." The judge will say, "No, we need DNA. We can in science we talk about DNA to prove that you are related to this person and all that." So superstitious and supernatural things cannot be scientifically proven. These claims are not able to be tested through variable observation. So it's not a scientific phenomena or scientific inquiry. What is suggested and testable explanation for an event called? A suggested and testable explanation. It's
A
easy,
hypo
guys. This one is easy. Come on. Well,
C.
somebody says C. Anybody else?
I also think it's C, sir.
C.
Thank you. You're correct. Remember, hypothesis needs to be testable. Let's go to the next one. What is the type of logical thinking that uses related observation to arrive at a general conclusion? We are coming to a nice stuff now.
It's A.
so I think it's B.
Okay, A and B. Anybody else?
I think it's C.
I'll also put C.
C, we got 2 C's now. And just expect to have a 2% or 5% when you're studying, that your examiner will deviate and ask something that he never said he will ask. So when you prepare and when you study, please go an extra mile. Don't just stick within the boundaries of the scope or the previous question paper or previous memorandum. Also go an extra mile. So here it's C, inductive reasoning, which takes specific data and uses it to draw conclusion about a general idea. I think last week when you are concluding, we also talked about inductive and deductive reasoning. And go back to those notes and check what is the deductive reasoning. So an example of how applied science has a direct effect on your daily life is the presence of vaccine that can contribute to individual and social health by preventing diseases such as polio, measles, tetanus, and even the influenza. Is this true or false? Anyone?
False.
Anyone? We've got true and false.
True, sir.
True.
It's true.
Yeah.
So can you please explain how? Isn't applied sciences more related to stats and risk management and stuff? How is it applied to a more biological sense of measles and this forth?
Yes, it's still an example of how applied science has a direct effect on your daily life. Has has an effect on your daily life is the presence of vaccine that contribute to an individual's social health by preventing diseases such as polio, measles, and before they arrive at the distribution of those vaccines, forget about the COVID vaccine, because yes, it might just spark another argument all over. But before they can distribute such vaccines, right? They first have to be people who did a research and who did the I think that's what the what do you call it? The people who were skeptical, the skeptics on the COVID vaccine, to say, you know what, there was not enough time to test and to have and inform research which was done to say this is the quality of this vaccine. This vaccine was not testable enough. It was not testable enough. So before they could do vaccine on people, on everyday people, or even on dogs, the first has to be the first people who going to do a research. After when they have done the research and do the testable and do the hypothesis and do the control experiment, they said, okay, this vaccine is fit for purpose now. It can be distributed either to people or other to these kind of animals. When we're talking about the veterinarians who will be like, the pigs, in order for the pigs to the farmers, in order for the goats to the farmers, the cows to the farmers, which vaccine should be used to prevent these such diseases. They first have to have a research guys who did an experiment. So this is an effect that this has a direct effect on our everyday lives, because before they distribute these vaccines, they first have to test them. So it's true. I don't know if I explained this enough.
Yes, you did, sir.
Yeah. So before something is being distributed, it first has to be tested. How is it tested? Through research. And for people to say this is quality, they have to compare it against the stats that they have done. What is the stats that they have done? Is that's why research is everywhere. It's in medicine, it's in economics, it's everywhere, because you're gonna have to collect the data and say, okay, this is the quality, this is what we need to do, and all of that. Yeah, but I'm glad that you asked that question, because some questions won't be like true or false. You might actually have to explain, why are you saying it's true? Why are you saying it's false? So this is the long answers where they are saying, although scientific method is used by most science, it can also be applied everyday situations. Think of a problem that you have at home, at school, within your car, and apply the scientific method to solve it. Anyone? We just did this. Anyone? All right, so this will be exactly what we did. You're gonna have to go for observation. Just think of an observation, then ask a question, hypothesize your observation, make a prediction. In that hypothesis had to be tested in a form of experiment, and then you're gonna have to do the analysis, and then you're gonna have to make a conclusion. In some ways, you can put your results and analyze those results and be able to come to an informed a conclusion. And this will be the problem. Just an example, my laundry takes long to dry the clothes in that order, and then you can do, I'm not going to delve much into this because it's the same example that we did previously. And give one example of how applied science has a direct on our daily life is the same thing that we're doing. So it will be, example is the use of smartphones on many scientific principle which include physics and optics, chemistry. We'll talk about battery, computer science, especially now computer science. Everything is softwares, whatever that you are using on your smartphones, all of those stuff. Name 2 topics that are likely to be studied by biologists and 2 areas of scientific study that will fall outside the realm of biology. This is just the topics. Biology topics. And the last question that a student asked, I think it's related to this one, but on a broad level now. You're just listing a topic which fall under biology and a topic which fall under outside biology, but they are both scientific topics. I think my research will be why are BLG 1501 male students not participating? That's my research. Does it has to do with Kaiser Chiefs and Manchester City not winning? I think that's gonna be my because they're blue ticking me today. Yes. But anybody with a answer?
I'm sorry, sir. I'm not getting this topic. Is it talking about topics that you can find or like research topics that someone can look into?
It's not just even topics, just general. Like when you're chapters, could be chapter, could be like a chapter, it could be like a, yeah, it could be like a, what do you call it? Chapter, topics, session.
I know. I'm thinking maybe human reproductive
Yeah,
systems.
you're right.
Human
Yes. Yes. So you can do like cell biology, studying the structure and functioning of cells, genetics or ecology, investigation, interaction, organism. These are topics. Think about the topics that we study about. There's a slide where we were talking about what is zoology and all of that. And if I didn't do that, I will also put it also in there. No. So outside biology, what will be your? It could still be your geology, which examine Earth's composition, and astronomy, which study all these topics which are part of science, but they may not necessarily be in biology. But that's what this question is entailing.
Okay.
Is it clear? Hopefully they don't bring this one in the exam. Thinking about topic of a cancer, write a basic science question and an applied science question that a researcher interested in this topic might ask. Any question. So here you're not going to be marked wrong. You just need to think about a scientific question that anybody can ask.
How
You
a
can.
cancer cell.
Okay, go.
Can you hear me?
Yes, I do.
Is a question like how are cancer cells formed?
Yes. Yes. Yes, you can use those questions like basic. So they could be basic. How do genetic mutation contribute to uncontrolled cell division in cancer? What are mutations, by the way? Those one who are doing BLG 1502, I think we did talked about mutations. What are mutations?
I think it is when when it alters, like I think it's the DNA of the cells or something when it alters in the nucleic sequences at all. I don't know how to properly explain it.
Mm-hmm. So this is like an error in genetic sequences, and mutations are the one that causes variations. So in your population or in your chromosomes or in the population. So when we talk further on when you go further in your second year or third year, they will be talking about the population structure and variation. So for a population to have clustering when you're doing those phylogenetic tree, their variations are caused by some deletions and insertions, which are the types of mutations, and those mutations can cause a bit of variations in your population. And the applied science will be, can we develop a drug that can target specific genetic mutation to treat a particular type of cancer in that order. Select 2 items which biologists agree necessarily in order to consider an organism alive for each given example of non-living object that otherwise fits the definition of alive.
Made up of cell.
Continue.
To be able to go through processes, I think biological processes.
So biologists will agree in things like metabolism, reproductions, where for metabolism will be like a fire consumes fuel and releases energy, and reproduction for which will be an example where crystal growing from a saturated solution which can replicate themselves. And here you can see even the made of cells, like you said, yes, and organized structure, which are living things that have a complex organization from a tissue and organs to an entire system. Non-living things, crystal, for example, which has a structured form, but lack the complexity of living organisms. Those are one of the examples. Another question, these are long questions, not multiple questions, which you're gonna be ready for. And yeah, please be ready for. Consider the levels of organization of the biological world and place each of these items in an order from the smallest level organization to most encompassing sequence. Cool. Sure. Go ahead.
Skin cell. Wait, hydrogen atom.
You're starting with hydrogen atom, and what else?
Then cell. And what? No, hydrogen atom. Water, because they're made up of atoms. Okay, atoms make elements which make up molecules. So it's gonna be hydrogen atoms, then water molecules, then skin cells, then liver, then elephant, then wolf pack because the community. Then what else?
Tropical rainforest.
Then planet Earth.
Okay. Congratulations. Yeah, I think you're right. Right. So it will be hydrogen atom, water molecule, skin, liver, elephant, wolf pack, tropical rainforest, and planet. Remember this diagram that we had previously, which talk about biosphere, ecosystem, organism, community, and all those stuff. This is the kind of questions that you might actually need to reference that diagram. Is it clear? Thank you. Then you go for a long walk on a hot day. Give an example of a way in which homeostasis keep your body healthy. We did talk about homeostasis, isn't it? Yes.
Sorry, sir, I was not here last week. Can you please give me a short description of what homeostasis entails?
Okay, not a problem. And anybody else? I will answer that. I will answer that when we are giving the answer to the question. I'm noting your question.
Since homeostasis
Mm-hmm.
also for keeping
Okay.
inside oh, okay. Let the guys answer.
No, continue. We'll go after you. It's fine.
Since it is responsible for keeping our body stable, I don't want to use the word stable, but like keep our body internal conditions not changing. Since you're walking outside and it's a hot day, your body outside, I mean the thermal temperature, it will hit on your skin, right? So our body, the metabolism that will happen is I think the blood. Okay, no, I'm confused. Let me let the guy speak, then I'll speak after him. I need to check something.
Yeah, you're confused. Very confused.
Yeah.
Okay.
Your sorry. Your
Yeah,
body
go ahead.
is hot outside, and your body is getting hot. So to try and cool the body down, you're gonna sweat, and that sweat is gonna make you cool down.
Okay, maybe let's first answer the first question. What is homeostasis? No, you don't need to worry about being, I almost say politically correct, being scientifically correct, just by your understanding what homeostasis is.
And homeostasis is the body's ability to try and maintain temperatures or
Thank you,
conditions.
thank you. It's about regulating. It's about regulating your internal body temperature in order to adjust your conditions so that you can be able to have optimal parameters for yourself to survive in each and every individuals. You remember we talk also about the invertebrate, the insects, how they practice the homeostasis. So homeostasis is about regulating your internal body temperature and maintaining a stability so that you can maintain optimal conditions so that you may be able to survive with the temperatures that are the changing temperatures. So when the temperatures are changing, now it's winter, you need to have means in which you can regulate yourself. When you move to a temperature are very high, you need to have means in which you can regulate your temperatures condition internally so that you can be able to survive. My guy, you said you still confused.
No, I didn't say I'm confused. I said you're gonna sweat. Then
No,
when
no,
you
not you. There's some. I'm talking about our friend here who only started attending today. Are you still confused? You're still not making any sense.
I'm getting there.
You're getting there. No, you'll get there. Don't worry about this. These slides, we're gonna share them as soon as we are done, and you can still go through them on your own. We're still gonna share these slides when I'm done with these sessions. So there's no cause to panic. So let's go to the answer. So homeostasis in action will be, for example, a way of maintain. Did I answer the question about what's homeostasis, my sister?
Yes, you did. Thank you.
Okay, thank you. Sweating, an example will be sweating is a way to maintain homeostasis, stable internal body temperature. And for example, like what my guy has explained here, you are very correct. Thank you, sir. During exercise or hot weather, your body produces sweat. As sweat evaporates from your skin, it removes heat, keeping your body cool. One way of your body reducing temperature is that you will see Most of the time, this is not a deep example. This is just a general observation and a general. Mostly, you will find that during winter, we release water by going to the bathroom a lot. We urinate a lot, and during summer or during hot temperature, people sweat a lot. Those who have dogs, who are dog lovers, you will remember, you will see during when the temperatures are very hot, they do, what did they call it? Is it patting? Dog
Yeah,
lovers?
patting.
Yes, where they will be like releasing a lot of, yeah, they will be releasing a lot of water and during the, as a way of maintaining their body temperatures. Do we have people who have dogs here? I'm thinking of getting a dog, but yeah, I'll see. Yeah. And using examples, what did I do? Explain how biology can be studied. I give the answer. Stupid, Murunga. What's the point now? Using an example, like explain how biology can be studied from microscopic approach to a global approach. If I were to say, did you know about this answer? I'm sure somebody will say, yes, I knew it, Murunga. Come on, I was going to say it. And I believe you. Microscopic approach and global approach. So microscopic approach will be biologists can study the cellular and molecular basis of life using microscopes and biochemical techniques. Then for the example, it will be to investigate the role of specific proteins and diseases. And global approach will be your biologists also study life on a larger scale, looking at ecosystem, biodiversity, and the effects of climate change on animal migration and patterns. What is happening now with my slide? Okay. Oh, we did this where we were doing the comparison of prokaryotic and eukaryotic. We're not gonna go there. Remember we did this about the phylogenetic tree. Just show with your thumb if you still remember. So we just want to do a recap. We just want to do a recap. So this is, take this as a recap. So I'm moving once. So we're doing a recap as well, which means I might not come back to this topic. But so take this. So this here will be a diagram with phylogenetic tree show a relationship of a domains of life. Don't forget about this. This is what we studied about. My guy who was confused, this is what we studied about. This is just a refishing. The domains of life is bacteria, archaea, and eukarya. And the first phylogenetic tree is a rooted phylogenetic tree, and the second one is an unrooted phylogenetic tree. Most probably one that we're going to talk about is the rooted phylogenetic tree, where it's got 3 branches where we're talking about the bacteria, archaea, and eukarya. We're talking about eukaryotes. And when in eukarya is where you find animals, fungi, plants, ciliates, flagellates, trichomonads. I'm butchering the names now. All these 3 names. And archaea, this is where we find all the halophytes. We're talking about halophytes, talk about Primordium, and we also talk about the T-cilia and all this. And for bacteria, you see about spirochetes, cyanobacteria, and apicloid and thermotoga and cytophaga, and all the gram-positive and gram-negatives that we did talk about, and the green filaments of bacteria. And so we talk about how we do writing about plagiarism. This is not my diagram. This credit to this, which is cited here also about that. So scientists use a tool called phylogenetic tree to show the evolutionary pathway and connection among organisms. So we did talk about a phylogenetic tree. What is a phylogenetic tree? It's a diagram to use to reflect evolutionary relationship among organisms or groups. And scientists consider phylogenetic trees to be a hypothesis. Remember, we're talking about hypothesis, a hypothesis of the evolutionary past, since one cannot go back to confirm the the proposed relationship. In other words, a tree of life can be constructed to illustrate when different organisms evolved to show relationships among different organisms. Like for instance, this figure, it shows to illustrate when organism evolved to show relationship among different organisms. A phylogenetic tree can be can be read like a map of evolutionary history, like a family tree. Where do we come from, and all those stuff. Many phylogenetic trees have a single lineage at the base, like this one at the base, and which represent a common ancestor. So scientists call such trees rooted. This one, rooted, which means there is a single ancestral lineage, commonly typically from down from the bottom left to the to which all organism represented in the diagram, which can relate. So if you notice that in the rooted phylogenetic tree, the three domains, bacteria, archaea, diverge. All of them, they have diverged from a single point or branch off, and small branch that plants, animals including humans occupy in this diagram show how recent and minuscule these groups are compared to other organisms. However, unrooted trees don't show common ancestors. This one does not show but show relationship among species. I don't think you need to worry yourself much more about the unrooted. It just indicates how related the species are. Then here's the question. Amy is studying this. Amy is studying. I'm sure those who watch The Big Bang Theory. Sheldon girlfriend, Amy Farrafella. Amy is studying phylogenetic trees of primates and their ancestors as part of one of her colleague courses. What information can she not find out by looking at phylogenetic tree? So we're checking what is the limitation of phylogenetic tree. Is it a timeline of when different species of primates evolved in relation to one another, or B, what chimpanzees have in common with orangutans, or C, what common ancestor different species primates share, or D, how primates are related to non-primates. So you are checking what phylogenetic tree cannot reveal, or what can she not find by studying a phylogenetic tree, by studying using these trees. So you're gonna have to go and check what does the phylogenetic tree entail. Remember we talked about phylogenetic tree. So probably most of these are what you can find using phylogenetic tree. So there's one option which you need to check with. Nah, man, no one can find with phylogenetic tree. What's the answer?
It's A.
A.
Did I hear A?
Yes.
Yes,
A. Because, yeah, it does not show evolutionary time constraints and all.
I think C, sir.
Anyone?
I think C.
Okay, anyone else? They so look the same. They so look the same. Okay. Anyone who? So it reflects. Phylogenetic tree can be read like a map of evolutionary history. So they have single lineage representing common ancestor. So the answer here is, excuse me. Sorry about that. B,
B.
because though phylogenetic trees can help to show ancestral relationship between organisms, it's not really specific enough to show how similar or different organisms are to one another on the before the branch level, before they have branched and all that. But I can argue it does show a timeline when different species of primates evolve in relation to one another, because those branches from one common ancestor is the regulation, or not regulation, is the time frame. Remember when we were talking about 100 million years back, where you concerned when they're referring to fossils and all of those. Is it fine? All right. So taxonomy, we did, my guy was confused. We did also talk about taxonomy. So taxonomy, which literally means arrangement, we are revising here, by the way, is the science of classifying organism to construct internally shared classification system which organism plays into one or more inclusive groupings. Think about how a grocery store is organized. Large space is divided into departments or compartments, such as produce, dairy, and meat. So when you go to the shop, wherever you shop, I'm sure it's a Woolies, not advertising them. I carry you guys have money. Obviously, there's categories there, all those stuff. Each department further divides into the aisles, then each aisle into categories on brands, and finally a single product where you can go say, I'm looking for a soap. This is the kind of soap, and this is exactly to the one that you're saying, I need this kind of. So this organism from a larger to smaller, more specific, it's called a hierarchical system. Remember, we did talk about something like this, where you find that the kingdom Animalia stem from eukaryote domain for the common dog classification level. Will be shown in this figure here. Excuse me. Therefore, the full name of the organism technically has 8 items. For the dog, it's eukarya, animal, chordata, mammalia, carnivora, canidae, canis, and lupus. Notice that each name is capitalized. Remember, we talked about the name. I'm talking to our friend here who started attending today. The name has to be italicized, except for species. And the genus and species name are, no, they notice each name is capitalized, except for species, and genus and both species names are italicized. If you are not italicizing and you forgot to italize, you need to underline. Scientists generally refer to an organism only by its genus and species, which is 2-word scientific name in which is called binomial nomenclature. Each species has a unique binomial nomenclature to allow for proper identification. And therefore, the scientific name of a dog is Canis lupus, and it is important that the correct formatting, which is capitalization and italics. If it's not in italics, you need to underline, which is used when calling an organism by its specific binomial nomenclature. So it will be a domain, kingdom, phylum, class, order, family, genus, and species. But you don't need to worry about knowing that maybe they will ask you the what is the scientific name or the order of this dog from domain to eukarya, but you just need to worry that it's domain, kingdom, phylum, class, order, family, and genus. Then we did talk about scientific inquiry. We just need to do a thorough recap on this one, because I got a sense that I think we didn't clear it enough about the inductive and deductive reasoning. So inductive reasoning is a form of logical thinking that analyzes trends or relationship in a data to arrive at a general conclusion. So the arrival or the conclusion is a general conclusion. Please underline that. A scientist makes observation and records them. These data can be qualitative, descriptive, or quantitative, consisting of numbers. And the raw data can be supplemented with drawings, picture, photos, or videos for many observation. So this will be like your scatter plots, your bar graph, your box plot, your bar graph, and all those, in order to represent or present your results. In other words, inductive reasoning involves making generalization from careful observation and the analysis of large amount of individual data points. So you make an observation, you do a proper research, but you arrive at a general conclusion. Generalization arrive at through inductive reasoning are not always correct. So their generalization or their conclusion, they are not always correct. Not making sense, right? Or making sense? Let's go to deductive reasoning. On the other hand, are you still guys hearing me? I can't see if I'm muted or not. Can you still hear me?
We can hear you, sir.
Sure. So deductive reasoning is another form of logical thinking that begins from general principle or a law, applies it to a specific circumstances or to predict specific results. So for deductive, you take a general information, and therefore you work on it to arrive at a direct conclusion. From a set of general principles, a scientist can extrapolate and predict results, specific results that will always be correct, as long as the general principles they start from are correct. So deductive reasoning and inductive reasoning move in opposite direction. Inductive reasoning goes from individual observation to broad observations. I hope it's starting to make sense now. While deductive reasoning goes from general principle to specific decision or prediction. Can you think of an example of inductive and deductive reasoning with these two statements? So inductive reasoning go from individual's observation to broad. Okay. So individual, your individual observation will be like somebody who you wrote a test. When you wrote a test, biology test, and that test more you. I'm sorry to use that term. And when it more you, and it's you individual, and you start to say, yes, that's biology is difficult. Biology will more you. Biology more people. Biology is difficult. When you see everybody who's working here who did biology, they were moved by biology. So you are making an individual observation and you are generalizing it. While deductive reasoning goes from general principle to specific principle, they say there's a general principle we say biology will more you, and you're like, I want to see myself if it will more me. And you go and enroll in biology and you don't study and it moors you. You're gonna go and say, okay, yeah, I had an experiment, it can. Or on contrary, you go and enroll, you attend this tutorial session, you start to study very hard and you arrive at a different conclusion. And said there's a general observation which says biology is difficult, but I attended it. It was never difficult on like that and so on. But the point here is that you move from general principle to a specific decision or prediction. Is that making sense?
Yes.
The difference between deductive and inductive. So please, whenever we talk about inductive reasoning and deductive reasoning, you should be on your toes. Again, inductive reasoning starts with individuals, pieces of information. Look for trends in the information. Twitter trends. Makes an informed guess based on those trends. Results may be incorrect. So for inductive reasoning, Results. These are the other description or differences. Results for inductive may be incorrect. This is an example of inductive reasoning. It's a nice food, eh? Last week, your best friend discovered a new restaurant that he really loves. He's eaten there 5 times already. In describing the restaurant to you, he says everything in that menu is delicious. You get him? So he went there, and last week your best friend discovered a new restaurant that he really loves. He's been there. He does have data. He does have data. He's been there 5 times. Okay, fair enough. He eaten 5 times, and let's assume he eaten 5 different menus. But in describing the restaurant to you, he says everything. Yo, Mngana, let's go there. Everything in that restaurant is delicious. He says everything. So this is an example of inductive reasoning. Your friend has eaten their 5 dishes at that restaurant. That's 5 individual pieces of information. 5. I'm sure they in their menu they have more than 30 dishes that they can do. They were all delicious. There's definitely a trend. The trend is that they are delicious. Based on his experience so far, he made an informed guess that everything else on the menu is delicious. But his guess may be incorrect because they might not be good in cooking spaghetti. They may not be good in cooking pap from us people from Venda. Is this making is this making sense now? You are understanding inductive reasoning. Let's go to deductive reasoning. Deductive reasoning starts with broad declaration like principles or law. Applies this generalization to make a decision about a specific case. Results are definitely correct. On deductive, results are definitely correct. So, for an example, driving. This is a general observation. Driving on icy roads is dangerous. Or driving, you don't have to use an American context. Driving while it's raining, it's very dangerous because the roads are slippery. The roads are icy today, therefore driving will be dangerous. So if it's raining, you will say the road is sleepy. I should have said, is it sleepy? Sloppy, right? Yeah. Why am I thinking of sleepy? Sloppy, because it's dangerous. That's everybody knows about that, right? You have heard somebody saying when it's raining, the roads are slippery, so it's dangerous. So this is an example of deductive reasoning. So we begin with 2 declaration about driving on icy condition or in rainy weather conditions. We derive a prediction about driving today based on the statements. The conclusions is very correct. Any question about deductive and inductive reasoning? You definitely gonna meet these questions in your exam or in your assignments. Please mention that. Any question? Those are clear, right? Good. I'm glad they're clear. There's a question. I carry you said they're clear. Police recently investigated a home burglary. DNA evidence at the crime scene allowed them to narrow the list of suspects to 2 brothers. Hey. After confirming that the first brother was out of the country at the time of the burglary took place, the police arrested the second brother. Which kind of reasoning did they use? Please also give a reason why you're choosing what you're choosing. Jesus, we're only left with 10 minutes. Yes?
Deductive.
Why are you saying deductive, sir?
Because based on facts.
Mm-hmm.
The fact that the DNA evidence at the crime scene
Mm-hmm.
pointed that the second brother is the one that committed the alleged crime because of the first brother was out of the country by the time the crime was committed.
The first brother was out of the country, yes. Anybody else? Wait, my guy, my friend there who was who's the first time is attending? Are you you think you can chip in on this one?
Hello.
You.
Yes. What was your question?
Do you think you can tackle the question on the screen here? Or is it making sense, the deductive and inductive reasoning?
Yes, it makes sense. You explain to us.
Okay. Do you think you can give the answer on the screen there? You can give it a try.
Someone else must try. I already answered number
You
A.
know what? Sorry, I didn't get
I
that.
answered letter A.
Okay.
Someone else must try. I must give others a chance to also prove their knowledge and understanding.
So
It's only
you're the
fair.
one who give the answer, right?
Yes.
Oh, sorry about that. Sorry about that. Okay. Anybody else?
So I would like to say it was an inductive reasoning.
Inductive.
A guessy prediction of elimination just because they confirmed that the brother was out of the country. They never of the DNA. They just guessed that if he was not there, then he didn't do the crime. Not really concluded on facts. So I think it was just a guess situationship.
Okay. Because of time, let's just go to the answer. But I like the reasoning, but don't worry about yourself. You will get there when you sit down with these notes, guys. And the police work from the set of known facts. Like what the first guy said, one of the first two brothers committed the burglary. One of the brothers was out of the country when the crime occurred. So they put this together to make their decision, which is a classic deductive. So it was a general conclusion. It was a general, when your observation, it will be like, okay, the other one was where? The other one was out of the country and the other one was here in the country. But they didn't just use only that. They infer the generalization with the set of DNA to say the DNA, it proves that the guy who was here in the country is the one that committed there, because the DNA still needs to match the guy who was in the country. So they move from general observation to specific conclusion. And this one, 3 years ago, a city announced a goal of cutting crime rates in specific neighborhood by 50% within 5 years. The next year, crime in the neighborhood was down by 15% in year 2. Crime had been cut by 28%. At the end of the year 3, crime was down 42%. The city just announced they are very confident they will reach the goal. What kind of reasoning did they use to reach this conclusion?
Inductive.
It's inductive, thank you. Correct, because they're starting from several data points which use inductive reasoning to use the trends of the data to make our best decision based on those trends. And that's it. Next week we'll be starting with the next session. And I hope this was helpful.